now we are going to start our today's class. And today we are going to discuss physiology of insulin, one of the pancreatic hormone. And you have already studied this hormone in relation to diabetes mellitus in your PBL case also. That will be now much easier for you to understand. And many of the aspects we have already understood. And try to make one more coherent view, more complete one. And many of the points you will come across that you have already studied, and try to make the whole one complete one. And it is also related to different pathological conditions, complications in diabetes also. The details you study in pathology, and the treatment. Again, insulin is also used as a drug for treating diabetes mellitus type 1 mainly, and also later in type 2 if necessary. Therefore, you will also study from the pharmacological viewpoint in pharmacology also, and everything will be correlated. I will here mainly focus on the physiological aspect. Okay, the outline for today's class will be introduction. You already know much of it. And mechanism of action means at the cellular level. Physiological actions. Then mechanism of secretion at the cellular level. Okay, and then physiological actions, mechanism of action. Whenever there is question about mechanism of action, first think of the events that occur when the hormone and the receptor interact. Think of where the receptors are located and how are they activated and what follows after activation of the receptors. Mainly we will focus there. And regulation of secretion, how insulin secretion itself is controlled. We know it is one of the major players in regulation of glucose and other pattern of utilization of energy, and its own secretion is also controlled to carry out that function. And we will see this one also. Okay, there are some questions for you to stimulate our discussion. Try to answer this one, true or false. Increase in insulin secretion decreases blood glucose level. All of you know it and answer it. Okay, try to participate in the discussion. If you write in the message, it will not cause any overload. Just give your answer. All of us can answer it simultaneously. There will be no overload in the system. I like to see all of you participating. Okay, it is true. That is the most basic thing about insulin. Insulin secretion rises after a meal that is rich in carbohydrate. Okay, good. That is also true. Next, insulin is essential to facilitate glucose uptake by the brain cells. True or false? Okay, false. Why is it false? You can speak also. Yes, glucose uptake by the brain cells is independent of insulin. Okay, what is the advantage of that? That independence of brain cells. Is there any advantage to that? Okay, good. Constant supply of glucose to the brain, even when there is. Okay, can you elaborate it? How brain cells they differ in terms of glucose utilization and dependence on insulin. Suppose there is insulin deficiency, and if there is one answer, brain uses ketone bodies as source of energy. Ketone bodies are not the first choice. When glucose is not available, then only ketone bodies can be used by brain to some extent. Mostly glucose is the first choice, and you can say solely. When it has to depend on ketone bodies, there will be many abnormalities in the body already. Okay, here we will come to it again, and to begin with here. There are two peculiarities of brain cells related to glucose metabolism. One is it depends almost solely on glucose for its energy, and another, it does not require insulin for glucose uptake. Therefore, even if there is insulin deficiency, brain will be able to use glucose. Ketone bodies are utilized when there is no availability of glucose. For example, in physiological one extreme, starvation, for example, that is not exactly physiological. Still we can consider it as at one end of the physiological variation. Next, glucagon is a hyperglycemic hormone. It means it will increase blood glucose level. Okay, good. It is true. In terms of glucose regulation, it has opposite effect as compared to insulin. Rise in blood glucose level stimulates glucagon secretion. That is false. The opposite is true. Now today we'll mainly focus on insulin, and in our next class glucagon also. Now we know the pancreatic glands are abdominal endocrine as well as exocrine glands. And here this microscopic figure shows different structures, histological features of the pancreas. And this one in the central, this structure is called islet of Langerhans. And the other structures here, these are pancreatic acini, acinus, plural acini. What is the difference, functional difference between these two structures, acini and islets of Langerhans? Which one is responsible for endocrine and which one for exocrine function? Now, most of the discussion here will be bilateral because we have already studied it in detail in your PBL class also, and you must have covered in anatomy also. Have you covered pancreas in anatomy? Yes or no? Okay, acini, exocrine, and okay, good. You have covered in anatomy. Acini, they have exocrine function related to digestion, pancreatic juices, and here we will deal with the islets of Langerhans, their endocrine function, and we are studying endocrine physiology, hormones. Okay. And within the islets of Langerhans also, there are different types of cells, namely alpha cells, then beta cells. These are beta cells. And the others, there are others also, delta cells. And you know their functions also. Which cells are responsible for secreting insulin? Beta cells. Okay, good. Alpha cells for glucagon, delta cells, somatostatin. Okay, most of the cells will be of beta type there, insulin secreting. Now, insulin, it is a polypeptide hormone. Have you studied insulin in biochemistry? Not studied it. Okay, biochemically, it is a polypeptide hormone. We know the different classes of hormones. And peptide hormones, we can say them peptide hormones, for protein hormones, or polypeptide hormones. As a category, they are same thing, though there are some differences from biochemical viewpoint. And being a polypeptide hormone, from our general endocrine physiology, we have described them, and they have different peculiarities. And you will note most of the peculiarities of the polypeptide hormones you will find with insulin also. And during its secretion, before secretion, during synthesis, there will be proinsulin, then proinsulin, and then finally insulin that is secreted. And this is the one diagrammatic representation of the insulin proinsulin molecule. And you can see here A chain, B chain, and C chain. And there are disulfide bonds connecting A and B chains. Okay. And before release, this pre-insulin is cleaved here, means degraded here and here. It means C chain is dissociated from A and B chains. A and B chain are still there. Therefore, insulin will have A and B chains both. this one C chain, and this one insulin, all A and B chains. And because this C chain was also a part of proinsulin, its significance is when insulin is secreted in the blood from pancreas, equal amount of C peptide will also be there in terms of their number of moles, number of molecules. If we measure concentration of C peptide and insulin in terms of moles per liter, there will be equimolar concentration. And its advantages: when we treat someone with exogenous insulin, we may not have idea how much the person is actually secreting insulin on his or her own. Therefore, to have the endogenous insulin activity, in insulin secretion, we can measure C-peptide, and that will be one indicator of the endogenous insulin secretion of the person. And insulin is transported unbound in plasma, and it correlates with the polypeptide nature of the hormone. If it was steroid hormone, for example, it would be bound with proteins. And plasma applies is short, and we know for polypeptide hormones it is shorter than the lipid soluble hormones. For example, thyroxine, it was in some days, and here it is in minutes. See the difference. And it has a lot to do. If we inject insulin and its plasma applied only 6 minutes, therefore we do not expect its activity to be longer lasting. Here I'm talking about the natural insulin, and there are different preparations that can prolong their stay in the plasma. Okay. Now, how is insulin secreted? I think you must have presented in PBL session. Can any one of us describe it? Sudeep?
Yes, sir.
Okay, can you explain this figure?
Yes, sir.
Okay, please try.
So, islets of Langerhans have GLUT2 receptors, two-way receptor. It sends the increased glucose concentration in the plasma. So, this increased glucose concentration via the GLUT2 receptor enters the cell, and this glucose is phosphorylated by glucokinase, and that leads to the formation of glucose 6-phosphate. And again, the oxidation of this glucose lead to the generation of ATP. This ATP will inhibit the ATP sensitive potassium channels, which lead to the depolarization, and this lead to the calcium influx. And this influx of calcium lead to the exocytosis of the insulin molecules from secretory granules.
Okay, excellent, very good. Okay, and now you know the mechanism, and here the main point. If you even forget the details, remember the initial stimulus. Increase in glucose will be transported into the beta cells. This one whole structure is beta cell in the islets of Langerhans. And here the glucose transporters are GLUT2, and there are different types. We will come to it again. And in beta cells, GLUT2 types are there. The glucose transporters there are of GLUT2, and they are they they transport by facilitated diffusion. Okay, it is passive mechanism. Therefore, depends on glucose concentration. Whenever there is increase in glucose concentration, there will be more entry into the cell. And finally, as Sudeep has nicely explained, there will be will be secretion of insulin. And here, remember these two, and never forget these two: increase in glucose concentration, increase in insulin secretion. And here, these vesicles, in the cells there are already stores of vesicles. Okay, therefore whenever there is sudden increase in glucose, they are rapidly mobilized, and exocytosis occurs that will immediately increase insulin secretion, and this response occurs within a few minutes. Therefore, whenever there is glucose concentration, increase in glucose concentration, there will be immediate increase in insulin secretion. And when these stores they are mobilized, after that the rate will decrease slightly. But again, there is new synthesis of molecules, and then the secretion continues. Now, once insulin is secreted, it will be transported in the plasma in the free form, and it races throughout the body by blood flow. Now, in the different target organs, and we know all of the cells will need glucose, but the mediation of insulin is not not equal for all of the cells. And we have already seen brain, it will not utilize insulin for regulating its glucose uptake. And many other cells will have effect, will show the effect. And most of them, most important of them are liver, skeletal muscles, and adipose tissues. They have very important role. Do not forget these three. All cells will be influenced by insulin except brain cells, but most important effect will be seen in liver, skeletal muscles, and adipose tissues. And you can see their importance. So liver has to do with storage. Skeletal muscles have to do with utilization. Skeletal muscle activity, that is one of the major way to expend energy. And then adipose tissue for storage of fats. Okay, here once insulin reaches the target cells, this is cell membrane of the target cell. And here insulin receptor consists of alpha and these beta subunits. And insulin molecules, when they bind with the alpha subunits, the intracellular portion will be phosphorylated. And here there is activation of tyrosine kinase enzyme. Therefore, insulin receptors, they are enzyme-linked receptors. We mentioned different classes of receptors. They are cell membrane receptors and enzyme-linked, because there is immediate contact with the enzymatic activity, tyrosine kinase. And this activation of this tyrosine kinase, it will phosphorylate different insulin receptor substrates. Means the substrates, various molecules, substances that are activated by or influenced by this tyrosine kinase. They are phosphorylated. And when we studied phosphorus, we mentioned its role, one of the important role in intracellular signaling also. Phosphorylation or dephosphorylation is the most common way to influence enzymatic activities inside the cells. Then there are a variety of responses, and one of them is glucose transporter, related to glucose transport. And another one, protein synthesis, fat synthesis, glycogen synthesis, and growth and gene expression. And here the different effects will take different level of time, different time period to show their effects. For example, glucose transport, it will be immediate within a few minutes. Protein synthesis, it may take some time. Fat synthesis also. And glycogen synthesis, relatively less. And there's growth and gene expression. And here we see that insulin has the effect on the nuclear machinery also, gene expression. Effects.
Sir.
This is the mechanism, and what are the final effects? As we have seen the different common of the effects. Now, what we will describe them. To summarize, insulin is an anabolic hormone, means it helps in synthesis of many macromolecules, and we can call it hormone of abundance, because when there is excess of energy available, excess of nutritional substances available in the body, this level will increase, and it is associated with abundance of nutrients or availability of nutrients. When there is large availability, it will mediate its effects. And it plays critical role in regulation of blood glucose level. We know it in relation to diabetes mellitus also. and utilization of various nutrients for energy, pattern of utilization of various nutrients for energy. In our last class, we discussed insulin acts as a switch between utilization of carbohydrates and fats or proteins. When there is increase in insulin level, carbohydrates are given the priority, while fats and proteins are spared or stored. And in the absence of insulin or low level of insulin, the opposite happens. Now we'll go to the metabolic functions one by one, beginning with carbohydrates. Insulin increases utilization of carbohydrates. When there is availability, good availability of carbohydrates, they will be prioritized and they will be utilized. And if there is excess that cannot be utilized by the cells, it will be stored. And mostly the storage form will be the glycogen in the liver and in the muscles mainly. We are talking about liver, skeletal muscles, and fats. And if you remember these three, most of the actions you will describe nicely. Glycogen in the liver and in the skeletal muscles, and fats in the adipose tissue. And synthesis occurs in the liver also, but finally the stores are in the adipose tissue. On lipid metabolism, insulin decreases utilization. See the opposite effect. Increases glucose utilization, but decreases utilization of lipids and proteins for energy, where utilization is for energy, and enhances the stores. And on protein metabolism, synthesis, somatic growth, decreased breakdown, and somatic growth, protein synthesis, and we know it is one of the important the mechanism for, for producing growth. Okay, this is the summary of effect of insulin on different metabolic aspects. Now, there are some other effects also. For example, on potassium transport across the cell, increase in insulin level will facilitate potassium influx into the cell. And so the final effect is decrease in potassium level in the plasma or extracellular fluid. And we utilize this property of insulin to treat hyperkalemia, short-term treatment of hyperkalemia. When there is very severe hyperkalemia, we infuse insulin, and that will transport the extracellular potassium to intracellular potassium. And finally, the extracellular potassium will decrease, and therefore many of the harmful effects of hyperkalemia can be for short term can be minimized. Okay. Now, carbohydrate metabolism. It increased insulin level decreases blood glucose level, and when there is decrease in insulin level, it will tend to increase blood glucose level. And we have seen increase or decrease in insulin synthesis is the major way to regulate blood glucose level. And the mechanisms, details of the mechanisms are promotes glucose uptake and metabolism by most cells, skeletal muscles, liver, adipose tissues, not forget. And in the liver, what happens? It promotes uptake, utilization, and storage. And the uptake, and there will be various enzymatic influences of insulin. I am not going into the detail of that. I think you will cover more details in biochemistry. And the glucose uptake is mainly mediated by induction of glucokinase activity. Means when glucose is taken up inside the cell, it will be phosphorylated. And now free glucose concentration inside the cell cannot build up. If free glucose concentration would build, the uptake would be minimized because there is passive transport. If there is no increase in free glucose inside the cell, glucose can flow continuously into the cells. And then utilization for energy and for storage also. Glucose, glycogen synthesis or glycogenesis. and it inhibits gluconeogenesis. Gluconeogenesis means synthesis of glucose molecules from non-carbohydrate sources like amino acids, for example. And you can understand it as there is already glucose excess in the blood. There will not be no need for synthesis of new glucose. Therefore, insulin will inhibit this one. That is advantage of this inhibition of gluconeogenesis, and promotes conversion of excess glucose into fatty acids. Glucose, when it is in excess, it will be first converted to glycogen, and glycogen stores they have also the limits. And when that limit is crossed, then the excess glucose that remains will be converted into fatty acids. And from the fatty acids, there will be synthesis of triglycerides and then VLDLs. And then those VLDLs, they are released in blood and they are carried throughout the body for utilization. And mainly the adipose tissues take them up. And the fatty acids from the VLDLs, they 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 cross the capillary membranes. And there is one hormone there. Do you remember the name of the hormone? Lipoprotein lipase hormone that will facilitate the uptake of fatty acids from blood into the adipose tissues. And hence you can see, you can correlate the facilitation for storage of fatty acid cells in the adipose tissue. We'll come to it again. And in skeletal muscles, there will be increase in glucose uptake by the action of insulin, its utilization again, and storage. It is similar. Overall, there is similarity with the events in the liver in terms of uptake, utilization, and storage. But the mechanisms are different here. Glucose uptake is mainly through the increase in number of glucose transporters. In case of liver, that was the enhancement of glucokinase activity. And regarding the storage of glycogen also, there is one major difference. From the liver, when there is low blood glucose level, the glycosenolysis will occur when there is low insulin level. But from the skeletal muscles, they can use the stored glycogen for themselves, but they do not release into blood. In this sense, we can say skeletal muscles are selfish. They store for themselves, not for the whole body. But by providing locomotion, they will help the rest of the body. Okay, brain cells. One another point for skeletal muscles. Though skeletal muscles they depend on, or they are in their glucose uptake is influenced by insulin. There are other ways also that enhance glucose uptake by the skeletal muscle cells, and it is exercise. With exercise, there is increase in movement of the transporters from inside the cell to the cell membrane, so that there are more transporters in the cell membrane, and there will be more diffusion of glucose into the cells. And this effect is not dependent on insulin. In skeletal muscles, insulin also increases the number of transporters in the cell membrane by moving the transporters stored in the cell into the plasma membrane. Exercise also causes the same effect. Therefore, exercise has one important role in prevention and also for treatment of diabetes mellitus. We tend to think that skeletal muscles, they increase glucose utilization during exercise, and that is the primary mechanism behind its beneficial role in diabetes. And the mechanism here is also the increase in number of glucose transporters in the cell membrane with exercise. And this effect is not only during exercise, it continues even after exercise is stopped. I mean, if we exercise in the morning, the effect will last throughout the day, for example. Therefore, regular exercise is important. Regarding the brain cells, we have mentioned Glucose uptake without intermediation of insulin means insulin is not the major factor to regulate glucose uptake by brain cells. And brain cells, they depend on glucose for energy. Many of the other tissues, with some exceptions of like retinal epithelium and in the gonad cells, and majority of others, they depend on different types of nutrients. When glucose is readily available, they utilize it. They give preference to glucose. But when glucose is not available, they can utilize fats also. And for heart, remember that even normal utilizes fatty acids more than glucose. Okay. Now glucose transfers. We talked about glucose transport, and there are different types of glucose transporters, and even the mode depending on the requirement of energy is different. One type is facilitated diffusion and another secondary active transport. Facilitated diffusion, and there are different types of glucose transporters that carry out that mediate facilitated diffusion. And there are different types of GLUT1. GLUT means glucose transporter. GLUT1 to 7F, there are different types. And here I have mentioned according to their importance. GLUT4, they are found in skeletal muscles, adipose tissues. And we know they are insulin sensitive. Means their numbers in the cell membrane, they increase with the level of insulin in the skeletal muscles and adipose tissues. And GLUT2, as Sudeep just mentioned, they are found in beta cells of the pancreas. And it has a role related to insulin. Means the beta cells, when there is increase in glucose level, they mediate the transport into the cell and then secretion of insulin is stimulated. And these other 1, 3 types, they mediate basal glucose uptake. Means even when there is no insulin or low insulin, these transporters, they will act as the baseline transport mechanism. For example, in brain and many other organs. And secondary active transport, it is not concerned with the insulin here. Just for your information regarding glucose transport in the intestine and kidneys, in the luminal site, these transporters are there. And on the basolateral membrane, again there will be other types of transporters, GLUTs facilitated, SGLTs secondary active, sodium glucose transporters. Okay, now effects on fat metabolism. Do you have any questions till now? Fat metabolism, overall there will be, insulin will facilitate stores, will facilitate fat stores, and different mechanisms are here. Lipid synthesis in the liver. Once glycogen synthetic mechanism is overloaded, there will be synthesis of lipids also, and that is transported to the adipose tissues to be stored in the blood through VLDLs, very low density lipoproteins. Synthesis of fatty acid in the adipose tissue itself. To some extent, adipose tissue can also synthesize fatty acid when there is increase in blood glucose level, for example, after meal. And lipolysis in the adipose tissues will also be inhibited, decreased. And that is mediated by hormone-sensitive lipase. That is increased, its activity is increased by cortisol, for example. Okay, hormone-sensitive. Hormone, that means that is not related to insulin, but other hormones like cortisol. Here, in fat metabolism, therefore, insulin will inhibit hormone-sensitive lipase, but it will facilitate lipoprotein lipase activity. Lipoprotein lipase activity is related to uptake of lipids, fatty acids from blood to the adipose tissues, but hormone-sensitive lipase is related to release of fatty acids from the stores, adipose tissue stores to the blood. Therefore, you can just predict the effect of insulin on those two different types of lipases. And One important correlation here will be in insulin deficiency. There will be excessive lipolysis and that will cause ketosis. When there is low insulin level or absence of insulin, there will be genesis of keto acids, mainly in the liver. In the absence of insulin, there will be increased availability of fatty acids. Those will be oxidated, and then different keto acids and ketone bodies they are produced, and that is the mechanism of diabetic ketoacidosis and can cause ketotic coma, and that is seen mainly in type 1 diabetes, where there is deficiency or even absence of insulin. And here the effect of insulin maybe seems to be mainly responsible, not only hyperglycemia, the effect of insulin, absence or presence of insulin. Therefore, that is not common in type 2, initial stages of type 2, when there is insulin level, adequate insulin levels. And in the long-term complication of diabetes is hyperlipidemia. And you can describe in terms of effect on fat metabolism. When there is low insulin level, there will be lipolysis, and it means there will be elevated levels of lipoproteins, lipids, cholesterol in the blood. And it is not only increased fatty acid level, it is also all of the different forms of lipoproteins except HDL. Now, effect on protein metabolism. Again, it is synthetic. It promotes amino acid uptake by the cells, just like glucose uptake. And there are various other effects also. And finally, there will be increased protein synthesis and inhibition of protein catabolism. Therefore, net gain, there is net gain of proteins. You can see the effect in a rat after depancreatization and hypophasectomy, means pancreas is removed and also the hypophysis and pituitary is removed. Now, what happens? We know growth hormone is a major player in growth, and we know insulin also is important for growth, and it shows what is the effect or interaction between growth hormone and insulin. You can see the rat here. It is depancreatized. Pancreas is removed and anterior pituitary is also removed. Therefore, there is no insulin, no growth hormone. Now, we inject growth hormone from outside and see the effect. The growth is not, the weight gain here is not so significant. Now we give insulin also and we see the effect. There is rapid increase in weight. It means growth hormone alone is not sufficient to promote growth. Insulin is also required along with. And we know the growth hormone, one of the intermediates are insulin-like growth factors. And it is easy to remember also insulin-like growth factors. They have similar effect on growth. And you can correlate with their function. Growth hormone also increases protein synthesis, and insulin also increases protein synthesis. Now, factors affecting insulin secretion. We have seen the effects of insulin on different metabolic reactions, different aspects of metabolism. You will study the more details of the enzymes in biochemistry. And here we will now see how glucose secretion itself is controlled. What affects glucose secretion? How does the beta cells know that insulin secretion should now be increased or decreased to mediate these different effects according to the exact need of the body? Now, there are some factors that increase insulin secretion and there are some that decrease. From physiological viewpoint, some are more important than others. For example, we have already seen increased blood glucose level will increase secretion. Similarly, increased blood-free fatty acids and amino acids will also increase. But the major regulator is glucose level, remember. We can forget others, but remember glucose. And there is one another interesting finding here. Gastrointestinal hormones like gastrin, cholecystokinin, secreted gastric inhibitory peptide, glucagon-like peptide 1. And these last two are also called incretins because they increase insulin secretion. And, and in our last class, we mentioned glucose intake by oral route has, has more stimulating effect on insulin secretion than intravenous glucose. And this finding finally led to discovery of these hormones. And when we take glucose or food by mouth, in the intestine, these hormones they are secreted, and they reach the blood. They reach through blood to the pancreas, and there they increase insulin secretion in response to increased glucose level. And this effect seems to be anticipatory. We can call it feed-forward mechanism because glucose level has still not increased to that level in the blood, but presence of food in the stomach, in the intestines, will increase the secretion of these hormones, and they predict that there will be this mechanism makes it sure that there is going to be increase in blood level in the blood glucose level, and therefore we require increase in insulin secretion. This mechanism can be considered one anticipatory or feed-forward mechanism. And other hormones like glucagon will also increase insulin secretion. Growth hormone and cortisol. With increase in growth hormone and cortisol level, there will be increase in insulin secretion. And the mechanism may be when there is increase in growth hormone and cortisol hormone, there will be increase in blood glucose hormone that may increase insulin secretion. And some of the factors that decrease insulin secretion are decrease in blood glucose level, opposite here, that is the most important, and others are fasting. Again, there will be decreased blood glucose level there. Somatostatin in the pancreas, one of the cells, D cells, delta cells, they secrete somatostatin, and that will decrease the insulin secretion in paracrine fashion. And leptin, another hormone from adipose tissue, that will also decrease insulin secretion related to obesity and so on. Okay, as we have already mentioned, glucose level is the primary controller. We can see here, with increase in glucose level, there will be increase in insulin secretion. And this increase in secretion is, the effect is more fast, more changed here after the glucose level 100. Below this one, there is no increase in insulin secretion. But after 100, that we take as the cutoff for normal glucose tolerance test also. After that one, there is rapid increase in insulin secretion that proportionates with increase in plasma glucose level up to certain. We can see here, even up to 200, there is already 10 times increase in insulin secretion. When this response is not very good or impaired, we call it impaired glucose tolerance test, because our body is not able to tolerate excessive glucose. And you can say it is a negative feedback control. Now, some questions for you. Any questions from your side? We'll try to complete in about 5 minutes now. Any questions?
No, sir.
Anyone? Okay, let's try these questions. Insulin increases glucose uptake by the cells by increasing the number of glucose transporters in the cell membrane. Okay, it is true. In many of the cells, that is the major mechanism. But in some cells, like liver cells, the enzymatic changes are also important. Anyway, overall, it is correct. And therefore, number of transporters is important here. Number, decrease or increase in numbers. brain cells use fatty acids as the primary source of energy. Okay, it is false. Next, exercise increases glucose uptake by muscles. It is true. And what is the mechanism? Mechanism of glucose uptake. Increase in glucose uptake during or after exercise, not only during, even after. Okay, translocation of GLUT2, GLUT4. GLUT4 means translocation means movement of these transporters from the cells, like inside the cells to the cell membrane. These transporters they are stored in the form of vesicles on the membrane of intracellular vesicles. And exercise will translocate the vesicles to the membrane and it will fuse with the membrane, finally increasing the number. And this effect of exercise is one of the important mechanism to regulate glucose levels. Okay, last, insulin secretion is increased by decrease in blood glucose level. False. It is simple. Okay, now it is simple for you, but with time you may tend to forget. And these simple basic concepts, if we do not remember this one, that is the problem. Okay, if we forget the more complex ones, that is not problem. There is another question. Hypoglycemic shock is caused by excessive plasma levels of, may be caused by, there may be other factors also, may be caused by excessive plasma levels of, okay, insulin. When there is increase in level of insulin due to some tumors of beta cells or exogenous administration, maybe as a part of treatment, when there is overdose, there may be hypoglycemia, and when severe, it can cause hypoglycemic shock. It may lead to unconsciousness and even death. And glucagon, it will not cause hypoglycemia, it will cause hyperglycemia. Polypeptide C, not much insulin-like activity, not, you can say, no activity. And cortisol, it will also cause hyperglycemia, not hypoglycemia. Glucagon is used for treatment of hypoglycemic shock. We will see it in our next class. The answer is insulin here. The stem was changed and there is no change in the answer. Here the answer is insulin. Okay, next we will study our another hormone from pancreas, glucagon, and briefly somatostatin also, and we'll try to correlate with diabetes mellitus also. Okay, any questions from your side?